I am Maheswaran. My project mates are Mithun and Kotteshwari. Our project topic is expression and characterizations of protease from Enterococcus hirae. Our internal guide is Mr. M. Chandran, Head of the Department of Biotechnology, VelTech, Dr. Rangarajan, Dr. Soundra, Engineering College. And our external guide is Dr. K. Srinivasabandam, working in a division of Bioproduct and Engineering in Pondicherry Center for Biological Research in Pondicherry. By this project, we identified that protease expressing gene from the Enterococcus hirae. Our aim is to identify that the specific gene that produces a protease in Enterococcus hirae. Enterococcus hirae is a gram-negative bacteria. It producing a protease, but it is pathogenic. It is not useful in commercial purpose. So, using a gene expression method, we collect the specific gene that produces a protease in the Enterococcus hirae and insert the specific gene into the non-protease producing organism and make it as protease producing organism. In this first step, we identify the specific gene that produces a protease in the Enterococcus hirae and insert it into the vector, and finally it is inserted into the non-protease producing organism and make it to produce protease. In our project, we use the E.coli as a non-protease producing organism. In the first step, we subculture the Enterococcus hirae from the mass culture in our institution, and we identify the protease activity by protein degradation method. By the protein degradation method, we can understand that the Enterococcus hirae is producing the protease. Then we move on to the primer designing. We design a primer by a bioinformatics method. Primer 3 is a tool used for primer designing. First, we retrieve the sequence, 16S RNA sequence from the NCBI and move on to BLAST for getting a similar sequence, and finally move on to the GLUSL-W to get a similar sequence in both the organisms to identify the specific gene that producing protease, and finally move on to the primer 3 tool to design a primer. By this way, we get two primers, forward and reverse primers. Forward and reverse primers that are containing a specific DC content. In this, we make a cage bait and stick out the E. coli, combinant cell E. coli into it. The protease is produced from the E. coli and degrades the casein presence in the plate. By this method, we can identify that the E. coli is producing protease. Thank you.